Hello. Hey, welcome, biology students. Hey, it is great to see you. Great to connect with you again. Looking forward to spending this time with you and for just sharing another great part of your educational journey. Again, thank you for opening this up and playing this and connecting in this way. As always, I'm always so excited about this stuff, and we got some really good stuff today. Active transport. Of course, this is part two of your plasma membrane series, and such an important organelle. What it does for us, what it allows for us to participate in, is phenomenal. But hey, as always. Hopefully you're doing well. Hopefully everything's going well for you, school-wise, family-wise, personal-wise, mental-wise, everything. We want the best for you at all times. Appreciate you guys, love you guys, and again, thank you for opening this up. So, hey, big stuff today. Active transport, and of course, you can see the stuff on the screen here on the right-hand side. That's for today. And you can see the stuff, just the edge of it over here on the left-hand side, right below the picture of the ogre there. But so that would remind us just to do a quick little review of what we talked about so far. Plasma membrane, highly selective, semi-permeable, which means it can control what comes in and out of the cell through recognition and through carrier proteins that are highly selective for particular things, and it's a great security check and balances for them. But we had diffusion, we had facilitated diffusion, we had osmosis, which is a huge topic, and certainly we worked hard to kind of work on that mental challenge of recognizing that it's water moving, but not focusing on just water moving for water moving's sake, but water moving with a purpose to dilute concentrations. So hopefully you were successful with that. So we've got everything we need. We got my notepad here full of all the good information. We got a glass of water. And so with that, hey, let's get started. So here we go. Now let's get our little text box up here, because we do have not a tremendous amount, but certainly some good information. We want to make sure it's nice and legible. So we'll be typing this out, and as you're writing it out, so let's get this started. Active transport. Okay, now active transport, we'll just get this right out of the way off the top of the bat, is the exact opposite of passive transport. So, if it's the exact opposite, passive transport did not require energy. So it's active transport. Energy is required. Okay, get that right off the bat. We're going to have to use energy, and we're going to introduce a molecule today, ATP, that we're going to talk about heavily when we get to chapter 6, chapter 7, chapters 8. They're just going to be very key. It's just a key molecule because it provides the energy that is needed for all this stuff to happen. Okay, again, if it's opposite of passive transport, it went down the concentration gradient. Now we're going to go up concentration gradient. Okay, we're going up. So if you can imagine pushing a boulder up a hill or walking back up a slide after you've slid down, passive transport, you'd be going down the slide. Passive transport, the boulder would be rolling down the hill. Now we're pushing the boulder up the hill. So that does require lots of energy. If you can imagine, remember as a little kid going down the slide, then turning around and walking back up the slide, That takes a lot of energy. So let me do a little drawing here. Let me close out of this. Now, actually, let's just slide over here. And where's that drawing? Okay, yeah, here's the drawing for passive. Okay, when we went down, we're going down the concentration gradient, and we're moving from where there's a bunch of it to where there's less. Okay, now we're going to do the exact opposite of that. And I'm going to get me a little ink here. Not the kind of ink that most people get, but something to write with here. And what would be a good color for passive transport? How about a nice orange? This orange right here. We're just going to call this red. No, wait, this would be the orange here. Yeah, so let's do that. So let me draw a little wedge here. And draw our little boulder here. And now we're going to go up. And if we draw our more or less gradient, more, less, that's supposed to be an E right there. We're going this way. And here's the way to think about that little metaphor here. I know it's rare for me to use metaphors, but here we go. And of course, that's a joke there. I use a ton of them. But think about a restaurant. Okay, if we were to go to a restaurant today, especially a very high level, not a high level as far as food, but a high volume restaurant. If we were to look in their pantry, okay, and if we were to look in their refrigerator, and if we were possibly to look in their freezer, we're going to find a lot of food. Lots of food. If we're going to look at the kitchen area, they're making lots of food. So there's a lot of food, whether it's being prepared right now or it's being stored right now. And stored is a great word for what we're talking about, because a lot of stuff is stored for later. But if we were to look at all those areas of a restaurant, we're going to find food everywhere, bunches of food, lots of food. But at the same time, there are trucks coming towards that restaurant, bringing more food. In other words, they're bringing food that they already have. They're bringing it to where there's already a bunch, and that's what we're talking about. And the reason why a restaurant has to do that is because not only does it got to make food for today, but it's got to be thinking about tomorrow, because it can't get up tomorrow and say, okay, we need to bring all this in, because then it's too late. Okay, if you need it now, you've got to already have it there. Okay, you can't get up, like an Olive Garden can't everybody come to work and say, all right, what do we need? All right, order it. No, it's too late. It can't get here that quick. So it's got to already be there. Already got to be en route. The process already has to be underway. And that's what we're going to be talking about today. Molecules that we already have in our cells, but we're bringing in more because we know we need them. And we can't wait until we need them then to try and go and get them. It's too late. The cell needs the energy, and so it's already got it. So basically, we're stockpiling. Okay, we're hoarding. We're bringing stuff in that we already have a bunch of because the cell knows it's going to need it, and it can't wait until it needs it to get it, to try and get it. It's got to already be there. And so that's what we're talking about. We're stockpiling resources. We're stockpiling ions, molecules, things that we need. Okay, constantly. We've got to have a constant stream of it coming in. And so that's what we are talking about today. So let me close out the ink tray. And I'm going to slide back over just a bit here. And click on this again. Edit, certainly. And continue our notes here. There are three types of proteins. I should say types of proteins. Classes of protein. Groups of proteins that are used, and I'm going to do this all in one line. Uniport, symport, and antiport. I'll talk that later. Okay, those are the three types. Oh, And I should have. Let me go fix that. That's going to bug me. Capitalize that. And now back down to my line here. Antiport. And we're going to talk about those. And you can see them right here. We're going to talk about them and talk about their importance and their direction and everything about them. Okay, another huge part of active transport is the sodium, potassium pump. I mean, it is a huge, excuse me, my eye is bugging me there. It's a huge deal, not only in general, but for instances like how the neurons pass information from one neuron to the next. This is a big part of that. So there's a lot of biological processes that use these pumps, and we're going to use the sodium potassium pump. Sodium Na is the symbol for sodium, and K is the symbol for potassium. In case you're wondering where I was getting those words from. Next up on the old list, coupled transport. Oh yeah. Coupled transport. That's a pretty cool process that the cell uses to sneak is a good word, but to get large molecules into the cell without requiring extra energy. But we'll talk about that. But it's basically a situation where we use some energy to move something across, and then while the protein is open, we bring in this larger molecule like a glucose at the same time to try to. And that's what the coupled is. We use energy from one process to get something else across. Coupled transport. Next up on my list of things that we are going to talk about, and I mentioned this in the opening, but ATP. That's the molecule that's going to provide the energy, and we're going to mention it that it happens now, and then when we get to chapter 6, the next chapter, we'll explain how it happens. But it's a simple process, and we're going to bring back old friends: dehydration synthesis, hydrolysis. Those are going to be key words. And so hopefully, way back in chapters 3 and chapter 3, that we got that process down. We just had a real good session with you Crowder students. Oh, that reminds me, I didn't do my welcome. Okay, let me finish this and I'll do that. But we had a pretty good session last Thursday with the Crowder students going over dehydration synthesis and hydrolysis. We had a good time. We had the chemical building blocks out, and we had the PowerPoint going, and we just had a real good powwow, just making sure that we understood that process. And we did the same. We would have done by this time the same thing with the high school students, because those two words, those two processes, man, they are so important. So let me back up, and since I reminded myself that I didn't formally greet. Hey, welcome, high school students at the Nevada High School, the bio dual credit people. Welcome, Crowder College students. Again, thank you for joining me in this way. And I've already mentioned this, but I'm just going to officially elaborate on it. But we're moving molecules and ions. I'm going to put it like that. Against concentration gradients. And just like I said over here, we are stockpiling. We're hoarding. Okay, we're bringing things in that we've already got. Because we know that we're going to need them, okay, and we're going to need a bunch of them, and we can't wait until we actually need them to order them. Okay, a company can't work that way, a restaurant can't work that way, a business can't work that way, and neither can the cells. And oh, this is huge. And I mentioned this, but we're going to officially write it down here. We use highly selective proteins. In other words, everything we talk about today, the proteins have to recognize what the molecules are, what the ions are. They have to physically latch onto the protein, be recognized, or else they don't come in. Okay, the stuff we're working with is big, and so it's got to have a huge security clearance. Just like as I mentioned last time, if you were at a high-end truck company, military base, whatever, a big important office complex where you approach and there's some sort of person at the front. Maybe it's at a gate, maybe it's at the desk when you walk in. Maybe if it's a truck company, you got to pass through a little security gate to where you have to tell them who you are, what you got, and they have to check on the list to see if you were expected to be there. And if you are not expected to be there, or you don't have authority to be there, then you don't get to come in. And that's what we're talking about here. These proteins have to recognize and to understand what's going on, or else it don't happen. All right, so that is the background information for what we're going to be doing today. So I'm going to center this up here, and let's get a little bit bigger here. And maybe I need to bring this down. So, well, you've written it down, so you've got all this up here. All right, so we're going to be talking about this scenario here. Now, this scenario is going to be kind of covering a lot of the other things we talk about. So again, it's background information, but it is information to make it easier to understand what we're talking about here. And let me get another little box here. That was almost well placed. So we're talking, and this is a huge science word, electrochemical gradient. Let me move that mouse there. And here's what we're talking about. The electro part, let me close this out because I want to underline these two words. So we're going to start with the electro part. In other words, electricity. And that's coming from these little symbols. Okay, those are ions. Okay, in other words, they've been a part of a bond, and they've taken electrons or they've lost electrons. If it's got a little plus sign, okay, if I draw, let me do it over here. If this is the periodic table, okay, the ones with the plus signs, they're on this side. That's the cations. Remember the cations from chapter 2? That's the cations, weak elements who cannot hold on to their electrons, and they've lost them. And therefore they have more protons than electrons, so that's why they got the plus sign. Okay, if they've got the negative sign, okay, then they come from over here. That's the anions. Remember the anions from chapter 2? They are strong elements, and they can hold on to their electrons, and yes sir, Bob, they will take yours if you do not watch it in an ionic bond. Okay, that's the electro part. And when we have these differences in positives and negatives, it creates a gradient. Just like with ions that want to come across, if they want to come across, and that's the chemical part. But just like diffusion, or if there's a bunch of sodium out here, it's going to want to go across the membrane, because you can see there's just one sodium here, and all these sodiums out here, they're going to want to go this way, okay, with diffusion. So electrochemical gradient means I had too many thoughts going there at once. So the electro part, if there's a bunch, so I know what I was saying. Just like with ions, when there's a bunch of sodiums out here, they want to go across. It works the same thing with pluses and minuses. If there's a bunch of pluses in here, okay, and there's very little pluses out here, the pluses and minus signs are going to want to go across. So it works the same thing with just the individual ions as it does also with the pluses and minuses. You can think of the pluses and minuses also as kind of entities that are wanting to balance out. You want to have the same amount of pluses and minuses on both sides. So if there's more or less, then they'll float in that direction. And that's the electro. And of course the chemical part is just the ions themselves. If there's a bunch of potassiums in here and only one out here, they're going to want to move across the membrane to balance it out. So the thing to remember is that both electricity, the plus and minus signs, the chemical, the ions themselves, everything wants to balance. Okay, everything wants to balance. And these proteins, these highly selective proteins that have to recognize things, are the gateways, the tunnels that is used to do so. So the take-home message here is that both the ions, the molecules, and the chemical symbols, the plus or minus signs, everything wants to balance. Okay, everything wants to balance. And part of how that gets done is that if you've got, for example, more positives in here and less positives out here, and you also have more potassium in here and less potassium out here, then the potassiums will move across, therefore balancing out the potassium and balancing out the plus and minus signs. So it's a pretty sophisticated, pretty elaborate process that the cell just does efficiently, effectively, beautifully, wonderfully at all times. So that's the electrochemical gradient. Okay, let me close up my ink tray here, and we're going to slide this up and talk about our three porters. Now, it's not a word that's used anymore, but for those that are older like myself, and you can recall a time back, especially in movies is where you'd see it, because I haven't lived a life where I've gone to a lot of big city hotels. I've been to a few through work-related situations, but now we call them bellman, bellhops, and I don't even know if those are words that are used anymore. But at one point in time, the word porter would have been used for those occupations, and it simply meant somebody who carried stuff, whether it was on a train or on a bus maybe, or in a hotel, just somebody who carried the bags, carried the stuff from where it was to where it was going to be. Okay, that was the word that was used. So that's the word, the background for that suffix there, porter. And let me get my pen up here. So that's the background for the word porter. Just a mechanism for getting things to where they are, to where they're going to be. And so now we have three types of porters. Okay, and the pictures are pretty self-explanatory, so I don't need to type or write a lot. But I will write a little bit, but I am just going to use letters. But I'll explain what I mean. So in a unit porter, okay, one molecule, and I'm just going to put the word M, one direction. I'm just going to use the word D just for space. So a uniporter, you can think of it as just a one-way street. Okay, we're moving one thing at a time. So if you think about a one-way street, there's usually just room for one car. You typically wouldn't have a one-way street that has multiple lanes, and maybe there's a big enough city where that is the case, but I've not experienced that. But we're moving one molecule, one ion in one direction. A symporter now, as you can see, can move two molecules in / ions. I should have probably done that, and I'm going to go back and fix that. Let me add that / ion, but also in one direction. So now we got a divided highway. Okay, think about the area that I'm in, Highway 49, Highway 44, or I should say Interstate 44, Interstate 49. Those would be good examples because they are divided and you've got two lanes. And so we're moving two things in one direction, as you can see in the diagram. Now the antiporter, now we're thinking about just a two-lane highway or two-lane road. And it may not, you may think that a highway is more important than just a road, and perhaps it is for volume, but in a sales situation, the fact that we can move two molecules or ions, but in two directions, in other words, at the same time, that's big, because that means if you use energy to get it open, now you got things that are going across both ways, and that's a pretty efficient use of the energy. So that's our three porters, and we mentioned those because as we talk about the figures down below or the images down below, we're going to be mentioning which class of protein we're using. Symporters, antiporters, that's the two we're going to mention. But we just talked about uniporter just so you have a point of reference that there is just one in one direction. So let me close out my ink tray here and move this up. And if you're wondering, man, how much more we got? Let's take a peek here. We've got 1, 2, 3, 4, 5, 6, 7 more images to talk about. But I will tell you those last 4 are super quick. And it's just one word and one note. And then boom, we're done, moving on. Look at me going side to side there. That's awesome. That's some good teacher skills right there. All right. Now this, and you can see our sodium potassium pump that we mentioned earlier. And actually that could stand to be a little bit bigger. Can't get my size thing to work. There we go. Alright, that's probably a little bit better right there. Alright. That wasn't so silky smooth. That wasn't great teacher skills right there. Alright, so again, plasma membrane. You can see potassium. You can see sodium. So sodiums in this story are the little 6-sided figures there. And potassium, I guess I'm going to assume are the ovals based on the colors here. Oh yeah, here it says sodium and potassium. There's their label. So here's what we got going on here. This is that sodium potassium pump, and so this is, and let me get me a little box here and put that. Let's go over here. So what we're going to talk about now is primary transport. This is a process under active transport, and there's a primary and a secondary transport, and it's part of that coupled transport process or thought process that I mentioned earlier, to where we've got to do something that requires energy, but as we do so, then we're going to sneak or we're going to move something else in the case that you'll see in a different protein. But the energy, we paid the bill, we overpaid. In other words, we had to break open an ATP molecule to release some energy, and the amount of energy that we needed to do what we were going to do is a lot less than what we were released. So there's some excess energy, so we're going to use that energy to do something else as well and not waste it. That's the best way of saying it, not wasting it. All right, so primary transport. We're going to move an ion across the membrane. Okay, in this case, it's either sodium or potassium. And using a uniporter, antiporter, we're going to do them both at the same time, okay, simultaneously. but let me make sure. Yeah, and so we're using ATP, and you can see that right here, ATP molecule, and you can see here when that separates, okay, when that phosphate is broken off, it releases a bunch of energy. And again, when we get to chapter 6, which is the next chapter, we'll explain how that happens and why that happened. Right now we're just going to reference that it is happening, or it did happen. Using ATP for energy. Okay, and when we move, and when this does happen, we're going to move, and I'm just going to put the word multiple because it's multiple sodiums or potassiums. Okay, we're going to move multiple of them at the same time, and you can see that happening here. You got 3 sodiums, you got 2 potassiums that are being moved across. So it's not one-to-one, and so we're talking antiporters here. Okay, we're using antiporters. Okay, so we're moving things in opposite direction. Okay, some are going in, some are going out, and it makes for a pretty efficient process. And just a reminder that we're going against the concentration gradient. We can't say that enough. Going against the concentration gradient. In other words, we're moving these ions from an area to where there's less to an area where there's more. In other words, we're stockpiling. Okay, we're hoarding. We need these elements, these ions. Okay, we need them, and we've already got some. We may even have a bunch, but we know we're going to need more, and so we gotta already have them. We gotta already have them there. Excuse me for a second. Getting a little dry there. Okay, against the concentration gradient, and these proteins that we're going to be using, highly selective. Okay, highly selective. They got to recognize the ions. They don't recognize the ions, then they don't come across. They've got to make sure that we're not letting something in that we do not need or do not want to be in there. So that's primary transport. Again, the take-home message is we're using ATP. Okay, we're using ATP to get this done. We are moving multiple things in multiple directions. Okay, but we also want to remember, because we're going to talk about it next, that the ATP molecule that was broken apart released, okay, a bunch of energy, and we can't get that back. So the cell says, all right, we've got this energy out there. We don't want to waste it. So here's what we're going to do. We're going to get this job done, and then we're going to do something else too. So we use that energy efficiently, wisely, fully. So let me close out of this and slide this. Oh, I didn't mean to do that. Slide this up. And now we're talking secondary. Move this over here. Secondary transport. Okay, we're talking secondary transport. And remember, this is a piggyback. Okay, this is a piggyback process. In other words, as I just mentioned, the energy that was used to get primary transport done is the same energy that's being used to get this done. In other words, like I mentioned, the energy that was released in that ATP molecule is more than what we needed. So the cell says, hey, we can't waste that, so we're going to do something else as well. And so here's what's going on. Secondary transport. Okay, we're using, and I'm going to put the word excess energy provided. Okay, I can't reference ATP enough. You got to have that in your mind. But we're using the excess energy provided by ATP. And what we're looking to do is move large molecules like glucose into the cell. The alternative to doing that would be to open up or break open the ATP molecule, release that energy, and move the sodium potassiums across as normal. Okay, and then also use another ATP molecule to bring glucose in, but that's wasteful because neither transaction would use the energy efficiently. So the cell says, no, we're not doing it that way. We're going to break open one, and we're moving everybody at one time. Okay, that's enough energy to get both jobs done. We're not going to have two separate transactions. We're going to have one transaction, two processes, boom, we're done. And so, but you can see here, and this is a pretty cool looking protein, almost like a Batman type thing here. But you can see the glucose, that's the big molecule here, and this is where the antiporter idea comes into play. And also, and I'm going to put this on here because it's very obvious in the picture, highly selective protein. And you can see that highly selective because their shapes, they got to match those shapes. They don't match those shapes, they don't come in. Okay, so again, the protein has to recognize the molecule, and it's got to be the right molecule. So we got these sodiums that are being moved in, and at the same time, we're going to slide this glucose in there. And this also is a reminder of stockpiling, hoarding. You can see all these glucose molecules in here already, and we're bringing more in. Just a reminder is we can't wait, the cell can't wait until we need it to go and get it. Too late. It won't get here fast enough. We won't have the energy we need. So it stockpiles, hoards, okay, bankrolls, whatever phrase you want to use. That's key. That's a key thing that we have to remember. So that's primary, secondary transport. Again, just a reminder, a review. Primary moves the ions across. It has to break open an ATP molecule, okay, to use that energy, but there's an excessive amount. There's leftover. And so the cell says, all right, since we've got that, we're going to do a second thing as well. We're going to get those glucose molecules in there, or any other or other large molecules. It's not just glucose, but this example we're using glucose, but it could be other molecules as well. And so that is secondary transport. Okay, let me slide this up. And we won't say much here because this is, and I will label it, but we will label it and introduce it, but we just talked about it, but this is the official name. Coupled transport. I'm going to put is primary and secondary. And what I mean by that, and again, let me make that, since I did the primary uppercase. Yeah, I know, it's crazy. That bothers me, but there we go. Coupled transport is the fancy name of that process, to where we break open ATP molecule, we do one thing, but there's leftover energy, so we do something else. It's coupled. In other words, the two transports are coupled together. They work off of each other. We do one thing, provide the energy, and because we got leftover energy, we do something else. So hopefully that makes sense. All right. So with that in mind, now we're down to these last 4 diagrams, and all we're going to do is label them and talk about them. I don't even need to. I mean, you'll write down what you need to write down, but I will just be drawing and talk about them, and they're pretty brief. It's really just to establish the scenario and what's going on. So we're probably less than 10 minutes. So here we go. Let me get this so you can clearly see the word. We're talking bulk transport. And I'm going to ignore my spider senses that are saying to capitalize that word bulk because it's the first word, but because we're moving on. And when I say bulk transport, here's what we're talking about. Sometimes the molecule that's got to move across is so large that we can't use traditional means. We can't use the regular way. It would just take too much energy. It would just take too much effort and time. They're just too big. Okay, just too big. So there are some things that the cell does in order to take care of that. And there are four ways, and each of these we're going to talk about each of the 4 ways. And the first one, so they're all bulk transport, and now we mentioned the name of this particular one, and you can see it. It's right over here, but I will also type it as well. And I drew a blank there, yeah. And it's pronounced phagocytosis, which is an unfortunate name, but that's what it is. Phagocytosis. And it's a pretty cool process. Now, all of them work in the same way. So once I described this, they all work in the same way. But what happens, let me close this out so I can get my pen here. But this, if you'll see this is the plasma membrane we're talking about. So this right here is the plasma membrane. And so what happens is this large particle, it approaches the membrane, and there's got to be some obvious security clearances. But we're not going to worry about that. We're just going to describe what happens. But what happens is the particle approaches the membrane, and the membrane, you can see here, begins to bend and basically just wraps around the particle. Okay, wraps around the particle, and then the part that is around the particle breaks off, and then the particle enters the cell, and now it's like a little vacuole. Okay, then, but if you can imagine at that point in time, we've got a plasma membrane that looks like this. Okay, that just joins together. That just comes back together. And whether it's a process where we bring in some more phospholipids and rebuild it, okay, however that process works, it just rejoins. I mean, obviously you can't have a gap in the membrane. It just rejoins, reconnects. So that's a pretty cool, pretty efficient manner, and just think your cell does that just without you even thinking about it. That's pretty awesome. But that's phagocytosis, moving large particles in just by budding around, enclosing the particle, budding off, and then just transporting it in to wherever it needs to go. That's phagocytosis. Okay, our next one, okay, and you can see the name right there, but I will also type it just so it's a little bit bigger and more clear. Pinocytosis. And the process here is whatever's coming in, it needs to be in a fluid environment. Okay, and so whatever's brought in also, we make sure we got fluids that come in with it. So it's similar to what we just saw, phagocytosis, but it's the incorporation of fluids as well. That's penocytosis. Okay, and again, here's the name, but I will type it just to emphasize it. But it's a name that is pretty self-explanatory. And then endocytosis. Now, close that out, get my ink up here. Now let's talk about this prefix here, endo, meaning you can refer to it like indoors, moving indoors. So we're taking something from the outside and bringing it in, but we're also using receptors that have to recognize the molecule or whatever it is. Again, we're talking large bulk, so we're talking big stuff. So whatever it is, it has to be recognized. And so these receptors are the same type of things that are used on the proteins to recognize things. So in this situation, again, the same thing. We butt around whatever we're bringing in. And so receptors and all, we all travel inside. We've collected it. We moved inside. Excuse me. And again, once that vesicle leaves, you got a protein or a plasma membrane that looks like this, and it just reconnects. Okay, receptor mediated. In other words, these receptors have to recognize, and then we just butt around, break off, and transport it inside. Bulk transport, big stuff, is the reminder here. And now, the last one. And it's key, and again, the name is here, but I will type it just to emphasize it. Exo. And hopefully that prefix jumped out at you, because the first three have been bringing molecules inside the cell. Okay, now we're going this way. Outside, exo, exiting. Okay, outside, getting it outside. So what we have in this situation is maybe a bunch of debris, waste products. We got to get rid of something. And it can also be that it's a useful something like a hormone, okay? Or maybe we've made a protein that needs to go somewhere else. We've made nucleic acid molecules that need to go somewhere else. It isn't just waste, but it could be waste. But the whatever's inside the vesicle needs to go somewhere else. And so now we're exiting the cell. Okay, but the process is the same. The vesicle approaches the membrane. Okay, the membrane separates, and this vesicle connects with it, and just simply releases the material. Now, the thing that they needed one more picture here. The picture they needed would be like this. The membrane has opened up, and I'm going to put the vesicle down here, and it's approaching. And when it gets there, and it's got all the stuff in there, I'm going to put the stuff in here, and it attaches to the membrane, and then all the stuff can then float out. That's the kind of, they got a before, they got an after, but they don't have a during. And so there's your during right there. The membrane opens up, the vesicle attaches, it opens itself up and connects to the two ends. It'd be like if you had say you need to get some water, you need to water a garden. It's a long ways from the hose, but you need more water that you can carry in a bucket. So you string out two garden hoses to connect it together. And when you're doing that, you realize, okay, that's not enough. I need a third hose, and you put the third hose in between the first two hoses. So you disconnect the two hoses, and then you add each of the ends of the third hose in the middle, and then you stretch it out even farther, something like that. I know it's been obvious to put it on the end, but that doesn't fit my example. Or that wouldn't have fit my example. So that is the idea of exocytosis. And that also brings us to the end of this video lecture over the plasma membrane. Unbelievably important stuff, cool stuff. Just, and your cell, I mean, I can't say it enough. What your cell does, what our cells do, is so efficient, so awesome, so wonderful, and the fact that we never have to think about that, and they're just remarkable. They're just remarkable things that what they can do for us, and the way they allow us to be such an effective working machine, and we don't even think about it. We think about the jobs that we got to have get done, while our cells are they're behind the scenes, just humming along, getting everything, making sure we got everything that we need. So, hey, again, thank you for tuning this in, for opening this up, playing this. And again, I hope everything's going well with you, with all that's going on. And just hope that you're safe and sound, and your family is safe and sound. And both physically, mentally, it's going well. I know the school year can be a trying time. And I've been there. You college students, I mean, even though it's been, seems like a couple decades, I can remember those days, the day-to-day grind, getting the stuff done, the memorization of just massive amounts of material. So I feel that pain. I feel your pain. And that's why I try to make myself as accessible and as helpful and as efficient as possible, because I remember those days. I remember the days at Missouri State, and it just so happened I had a Missouri State shirt on today. I can remember those days at Missouri State, and of course when I started there it was Southwest Missouri State. But I can remember those days when you end your finals and that last one is taken and you just want to go somewhere and sit down and just relax, and just you got that feeling of accomplishment, but you've also got that feeling of, oh man, it's done. And then you can begin to relax and unwind. So I know what you're going through, and anything I can do to help, you just let me know. Let me know how, when, and where, and we'll make it happen. So with that, that's enough blubbering by me. We're going down the sentimental journey there, because it is a special time. It was a special time for me. I cherished it. It was hard. It was difficult. It was challenging. But I loved it, and still recall those memories, still recall the people that the instructors that I got close to, just because I needed them, because they were my bio people, and that was my field. But anyway, with that, hey, let's say goodbye until next time.